Chapter 6. Techniques of Genetic Engineering. Success in genetic engineering has been possible due to rapid development in gene cloning methodologies. It is essentially the insertion of a specific fragment of foreign DNA into a cell through a suitable vector, in such a way that inserted DNA replicates independently and transferred to progenies during cell division. There are many techniques for artificially introducing genes into the recipient organisms, such as vector-based technique called recombinant DNA technique, and the 6-Tahar techniques such as electroporation, microinjection, bioballistics, etc. The transformed cells containing DNA after their characterization and conformation can be used commercially for the production of useful compounds such as insulin, interferon, growth hormones, etc. A gene cloning in prokaryotes. The word cloning refers to the methods involved in replication of a single DNA molecule starting from a living cell to produce a large population of cells containing similar DNA molecules. Generally, DNA sequences from two different organisms are used in molecular cloning, one that is the source of the DNA to be cloned, and the other that serves as the host for replication of the recombinant DNA. See the figure. Structural organization of a typical gene construct. See the figure 6.1. Diagrammatic representation of gene cloning. The chemical structure of DNA is fundamentally the same in all living beings. Therefore, if a DNA segment from any organisms is inserted into a DNA segment containing DNA replication sequences, and the resulting recombinant DNA is introduced into the host cells, the DNA insert will replicate with the DNA of host cells. The DNA fragment to be introduced into the host cell is called insert DNA, desired DNA, target DNA, or foreign DNA. Such gene DNA fragment is taken out from an organism or gene library, i.e., a random collection of cloned DNA fragments in vectors that consist of all genetic information of that organism concerned. However, getting a desired DNA is not so easy because the cellular DNA is very large and the position of desired DNA is not accurately known. Libraries of such cloned fragments are prepared. B. Strategies of recombinant DNA technology. There is no single method of recombinant DNA technology, but it involves several steps. In 1976, Methodis and co-workers have described the basic techniques of gene cloning. The principal steps of gene cloning are shown in figure 6.1 and given below. 1. Isolation of DNA of known function from an organism A. 2. Enzymatic cleavage B, and joining C, of insert DNA to another DNA molecule cloning vector to form a recombinant DNA, i.e. vector plus insert DNA, molecule D. 3. Transformation of a host cell, i.e. transfer and maintenance of this rDNA molecule into a host cell A. 4. Identification of transformed cells, i.e. cells carrying rDNA, and their selection from non-transformants. 5. Amplification of rDNA F to get its multiple copies in a cell. 6. Cell multiplication G to get a clone, i.e. a population of genetically identical cells. This facilitates each of clones to possess multiple copies of foreign DNA. cDNA, gene library. A gene library is the collection of different DNA sequences from an organism where each sequence has been cloned into a vector for ease of purification, storage and analysis. There are two types of librands on the basis of source of DNA used, one genomic library where genomic DNA is used, and 2, cDNA library where cDNA or complementary DNA is produced using mRNA. A gene library should contain a certain number of recombinants with a high probability of containing any particular sequence. This value can be calculated if you know the genome SU and average size of insert in the vector. The probability of getting a given sequence in a gene library, the number n of recombinants, bacterial colonies or viral plaques, can be calculated by using the following formula, where p equals the desired probability and f equals the fraction of genome in one insert. For a probability of 0.99 with insert size as 2x 10 to power 4 BP, the value of genome for E. coli 4.6 into 10 to power 6 BP will be. See formula. 1. Genomic library. It is a collection of clones that represent the complete genome of an organism. All fragments of DNA inserted into vectors for further propagation into suitable host represents the ensure of an organism. For construction of a genomic library, the entire genomic DNA is isolated from host cells' tissues, purified and broken randomly into fragments of correct size for cloning into a suitable vector. There are two basic ways of fragmenting genomic DNA randomly: physical shearing, e.g., pipetting, mixing or sonication, and partial restriction enzyme digestion. By using limiting amount of restriction enzyme, the DNA is not digested at every recognition sequence. Using these methods, genomic DNA is broken randomly into smaller fragments. Figure 6.2 shows a total of 6 random DNA fragments obtained after physical shearing. The vector isolated from bacterium is also digested with the restriction enzyme which digests genomic DNA. The fragments of genomic DNA is inserted into the vector. Each vector consists of different fragments of DNA. The recombinant DNA molecules are transferred into bacterial cells or bacteriophage particles are assembled. The genomic library for organisms with smaller genome size, e.g. E. coli, can be constructed in plasmid vector. Only 5,000 clones, of the average size of 5,000 BP, results in 99% chance of cloning the entire genome of 4.6 into 10 to power 6 BP. In addition, libraries from organisms with larger genomes are constructed using phage lambda, cosmid, BAC vectors. See the figure 2. cDNA library. The mRNAs are highly processed representatives of genes which express under specific conditions. The mRNAs cannot be directly cloned because they are unstable, hence mRNAs are convened into cDNAs. The library made from complementary or copy DNA, cDNA, is called cDNA library. The cDNA library represents the DNA of only eukaryotic organisms, not the prokaryotic ones. Because genomic DNA of eukaryotes contains introns, regulatory regions, repetitive sequences. Therefore, establishment of genomic library of eukaryotes is not meaningful. The cDNA library can be constructed by using mRNA because mRNAs are the highly processed, intron-free representatives of DNA having only coding sequence. Synthesis of cDNA has been given in Figure 6.3. Figure 6.3. Synthesis of cDNA by using mRNA. Keeping in view the aim of the experiment, DNA molecule is either enzymatically fragmented or may be procured from cDNA or genomic libraries. Getting a cDNA fragment of its known function or characterizing its sequence is very difficult. A cDNA fragment is prepared directly by using mRNA as template. It follows many biochemical methods. Necessary enzymes with their restriction sites and cleaved products required during the isolation of DNA have been described earlier, and the important enzymes are given in table 4.1. A. Isolation of mRNA. It is much difficult to isolate a specific mRNA if it is in low amount in the cell. The majority of mRNA sequence in eukaryotic cells contains a long polyadenylated tract, i.e. about residues, at their 3' termini. Cates, 1970. By this virtue they can be separated from other types of RNAs. Therefore, mRNA binds with an oligo-dt cellulose affinity column or poly-U cephalose from which it can be eluted. Its integrity can be studied using gel electrophoresis, which allows mRNAs to be size fractionated by recovering chosen regions of the gel lane. Commercial preparations of both these substrates are now available. The mRNA preparations can be enriched for the desired molecules by fractionation according to their size by using sucrose density gradient centrifugation. Preparation of cDNA is shown in Figure 6.3. B. Reverse transcription. Reverse transcriptase is required for the synthesis of DNA copy of an mRNA molecule. The mRNAs are treated with oligo-dt primer, reverse transcriptase enzyme, and 4-deoxynucleotide triphosphates DNTPs, i.e. DTP, DGTP, DCTP, and DTTP. The oligot primer binds to polyadenylated tail that contains quadruple A end residues and provides free O site for reverse transcription. Reverse transcriptase adds complementary DNTPs 1 by 1 to free 3' O site of primer, forms single strand of DNA resulting in RNA-DNA hybrid. Cellular DNA and total RNA inhibit reverse transcriptase activity. Therefore, it is necessary that mRNA must be in pure form before cloning. The oligo-dt contains 10 to 20 nucleotides that hybridize the poly-A-tract on mRNA. The oligo-dt primer ensures the initiation of cDNA synthesis at 3' terminus of RNA in poly-A-tract. C. Oligo-dC tailing. The RNA-DNA hybrid is treated with the enzyme terminal transferase and a nucleotide precursor, DCTP. This enzyme adds DCTP 1 by 1 to 3' O group of RNA and DNA strands. Consequently, a short oligo-dC tail is produced at 3 feet end of both the strands. The residues of cDNA become curved. The curved tail forming a loop-like structure is known as hairpin loop. D. Alkali hydrolysis. Upon hydrolysis by using alkaline sucrose solution, the mRNA cDNA strands are separated into single strand. A. Addition of oligo-G primer. Oligo-G primer is added in the reaction mixture and temperature is maintained to about 550 C. This facilitates the binding of DG to oligo-dC tails formed on cDNA. Now cDNA acts as a template for the synthesis of double-stranded cDNA in the presence of DNA polymerase I AMV avian myeloblastosis virus reverse transcriptase. F. Synthesis of second strand of cDNA, DNA polymerase I and dNTP precursors are added to the reaction mixture. This enzyme adds complementary nucleotides to the cDNA 1 by 1 to 3 feet or site of primer. Consequently, from one cDNA, double-stranded DNA clone is synthesized, the length of which is similar to that of the mRNA molecule initially used. This is used for cDNA cloning in construction of cDNA library and in gene cloning experiments. The double-stranded cDNA is identical to the gene which codes for mRNA only in prokaryotic genes. However, the majority of eukaryotic genes contain introns, the non-coding regions, which interrupt their coding sequences. Methods of removing cDNA in eukaryotes have been described earlier. cDNA can be radioactively labeled by nick translation and used as a hybridization probe for the identification of DNA fragments containing the gene. By using these procedures, a cDNA clone bank is built up. See preceding section D. 3. Screening of cDNA clone. Since cDNA clone needs prepared by using a specific mRNA, there is no need of screening the cDNA clones. However, often cDNA clones are the mixture of cDNA molecules. The cDNA clone of desired function may be low or large in number. Therefore, all these clones must ligated to a suitable vector such as Me3 vector, phage vectors and transferred into host B. Each bacterial cell possesses a single cDNA clone, hence collection of all recombinant B is called cDNA library. When gene cloning needs to be done, the insert DNA is isolated from recombinant bacteria and cut with restriction enzyme. Then the cDNA is separated through electrophoresis and used for gene manipulation. D. Insertion of a foreign DNA fragment into a vector.